Alright, so this is part 2 of your evolution video notes, and this set of notes is going to talk a little bit more about the evidence that exists for evolution. So there's 4 things total. You have your fossil record, anatomical evidence, which is like structural, molecular evidence, which is more of the DNA level, and then embryonic development. If you remember, the embryo is the growing baby. So fossil record. Why are fossils important? So 2 reasons. They are a record of an extinct species, and they determine the ancestry of an organism. And there's 2 types of dating. So you have relative dating and absolute, or also called radiometric dating. So the fossil lab you did in class was a relative dating. You were determining the approximate age by looking at their location in the rocks. Absolute dating uses actual isotopes to measure the exact age. Okay? If you go into a class that talks about more of the Earth systems and fossils and rock layers, you'll get much more into absolute dating. So anatomical evidence is the second thing, and there's a couple terms that should be familiar to you. So the terms homologous and analogous. But there's also something called a vestigial structure. So let's quick review. Homologous, if you remember, homo means same. So they have the same structure, same bones, same parts, but they have a different function. So the best example of this is a whale fin versus a bat wing, or any other mammal for that matter. You could say versus human arm versus cat leg. They all have the same bones, but they have different functions. Whales swim, bats fly, humans pick things up, cats walk. Okay? Your analogous structures then are the opposite. They're a different structure, but the same function. They are not from a common ancestor. There's butterfly wing versus bird wing is our example here. So butterflies and birds both fly, but butterflies are insects and birds are vertebrates. So big difference. The last thing is a vestigial structure. So this is the reduced form of something that used to be functional at one point for the organism, but is no longer functional. So for example, humans have an appendix, but we don't need it. Their appendix will burst. Some people have them removed. It's not necessary. Whales have a pelvic girdle. Pelvic girdle, that's like your hips, where your legs would come from. Well, whales don't have legs. They have their fins, but they don't have legs. But they have the pelvic girdle that shows that their common ancestors at one time had hind legs, which is where the idea that whales' common ancestors walked on land came from. So here's just an example then of your homologous structures. So you see the human, the cat, the whale, the bat, they all have the same bones. They're color-coded. They all have the orange bone. They all have the yellow bone. They all have the cream-colored bone. They all have the brown bones. All of them have that same bone. It just has different shapes and different functions. Analogous structures, on the other hand, like we said, are the same thing, but they're different. So this shows a bat versus a bird versus an insect. A bat's a mammal, a bird is a bird, and then an insect is not even a vertebrate, but they all help them fly. And then here's an example of a vestigial structure. So it actually shows you how there's the femur and the pelvis that exist in the whale, but they don't function for it. So they don't have a function, but at one point in time they used to. Now we get our molecular evidence. So the more similar the DNA, the closer the relationship. That should be a given. That should be something that you assume always. But proteins are also similar between recently involved species. So for example, dinosaurs and chickens have very similar protein and DNA structures because birds and reptiles evolved very close from a common ancestor compared to the other major vertebrates that we think of. And last is embryonic development. So a similar development suggests that they're more closely related. If you look down here, if we kind of try to find the mammals, so mammals are on this end. So you have a human, a rabbit, a cat, a hog. So those are all the mammals. They all look pretty similar, right? Then you have a chick, a salamander, or a tortoise, sorry, a salamander, and a fish. So if you look, you can kind of see how they're more closely related. So reptiles and birds are more closely related, as you see that. Fish and amphibians are more closely related. But at the same time, all of these have a similar embryo development, which is why they're all considered a vertebrate. That's their common sharing that they do. So now on to shaping the evolutionary theory. What are some things we see in nature, things we see that would shape this theory? So there's a couple effects or things that can happen. First is the founder effect. A new population is established by a very small amount of individuals, so a founder. If someone's the founder of something, they started it. But when you only have a few people that start something, then there's very little genetic diversity. As a result, if there's a defect or something wrong with someone, like this guy, it shows up a bunch more generations later than it would have had he stayed in the original population. There's also something called the bottleneck effect. So if a large percentage of a population is killed off and only a few survive and then continue to reproduce, you only get certain traits. So if you look, and as it says, usually occurs after a natural disaster. So if you look, remember the bees, those symbolize the genes. So lowercase is recessive, uppercase is dominant, and then heterozygous. Well, if you look, there was some sort of natural disaster, all these dominant alleles are gone. So look, your resulting population is made from recessives. Same thing, look over here at this diagram. You have all these different colors present, but then an event happened, and over time, look what we have. Just the two colors left. There's also something called gene flow. This is just genes that enter or leave a population. Most commonly is migration, some sort of migration. So emigration with an E is when you leave. Immigration with an I is when you enter. So if birds enter, they have babies, that enters, that's considered an immigration. A death is leaving. And also emigration is if birds decide to leave that population. So birth and immigration increase genes, death and emigration decrease the genes. So you want to have that constant gene flow to keep your population diverse. The next thing is coevolution. So coevolution is the evolution of one species, but it directly affects the evolution of another. So if we look at this picture here, as the flower evolved a longer and longer tube, this insect evolved a longer and longer nose to get to the pollen in the flower. So they are co-evolving. They're always going to evolve with each other because they depend on each other to survive. Same thing with this here. This insect looks like the leaf, but if the leaf were to change, then the insect would also change to reflect the leaf. Then we have convergent evolution. So convergent is when unrelated species evolve similar traits, but they still live in different parts of the world. So if we look, it's kind of similar to your analogous type of structures. So they do the same things, but they live in different parts of the world. So they're converging, they're evolving together, but they're in different sections. Divergent evolution, on the other hand, is where one species divides into two and they become more and more different. So divergent, you're diverging, you're separating. So if you look, this was our original butterfly. We have one type going this way and becoming more prominent, another type going this way and becoming more prominent. And then really quick, we're going to do a cladogram practice. So we did this one in class. You want to figure out what letter goes where. So first of all, let's start, let's do something easy. So curly antenna. Who has curly antenna? The only one with curly antenna is this guy. So H would go there. So we have that one. Okay, next we have, let's look for something simple. So 6 legs. Well, we know spiders don't have 6 legs. They have 8, and this guy has no legs. So 6 legs is going to go at C. Wings. Where do wings start? Wings look like they start at F. A segmented body. So a body that's broken into segments. Well, the spider is, but the worm isn't. So segmented body is going to be B. Double set of wings, that looks like it starts at G. Jumping legs, that one's pretty specific. That's just going to be E. Crushing mouth parts, well, oh, didn't realize that. Whoops, we're going to go back. So segmented body, I guess we're going to consider this guy as having segments. So segmented body is going to be A. And then legs is going to be B. Crushing mouth parts is going to be D, the ant. There's another way to do a cladogram. So when you have animals on the table, you use these characteristics. So let's see, does a slug have cells? Yes. A backbone? No. Legs? No. Hair? No. Thumbs? No. Does a catfish have cells? Yes. Backbone? Yes. Legs? Nope. No hair or thumbs. Frog? Cells? Backbone? Legs? But that's it. Tiger? Cells? Backbone? Legs? Hair? No opposable thumbs. And humans? Check all 5. So then what we would do is you would make a cladogram. So we're going to go back and forth here a little bit. So we're going to draw one. Okay, so first we have a slug, then a catfish. We're going to do slug, catfish, frog, tiger, human. Okay, so if we go back and look at our characteristics, cells, well, they all have cells. Let's see. From catfish on, they have a backbone. From frog on, they have legs. From tiger on, they have hair. And from human on, they have opposable thumbs. And as you know, as we go up, it's a more and more recent species, and there's more and more diversity. Okay? And that's it for your notes.